Rappin with ReefBum is sponsored by Bulk Reef Supply and Ecotech Marine.
Hey, what's happening, everybody, and welcome back to another episode of Rappin with ReefBum. I'm your host, Keith Berkelhamer. So today on the live stream, I welcome Kenneth Wingerter from Hydrospace LLC. What's happening there, Kenneth?
Hello, everybody.
So Kenneth is a lifelong aquarist with diverse interests ranging from cold water, marine aquariums to ponds and, all right, I'm gonna mess this up. Paludariums? Is that how you pronounce that? Paludariums?
Paludariums.
Paludariums.
Paludaria,
Okay.
if you want to be
Okay.
ultra technical.
Okay. Since completing studies at the University of Oregon and the Aquarium Science Program, he has practiced aquaculture in laboratories, hatcheries, with a focus on live microfeed. He currently resides in Colorado, where he operates the microbial aquarium product company Hydrospace LLC. But before we start chatting with Kenneth, I want to thank the sponsors for the live stream, both Bulk Reef Supply and Ecotech Marine. I really appreciate these companies supporting the live stream. And I also appreciate all you folks out there that are tuning in right now for being in the stream. And as always, please drop your comments and questions in the chat. Try to work those into the conversation. So, Kenneth, man, your company is pretty well known in the hobby for selling the microbial products, especially the purple non-sulfur bacteria. Can you just give the folks that are not familiar with the company a broad stroke overview of what you guys are all about and why you started the company?
Yeah. First of all, it's all we sell right now is purple non-sulfur bacteria. I'm not sure if I'm going to wander outside of that, or at least outside of the realm of photosynthetic bacteria. A person could actually spend their whole lifetime studying these organisms. They're so complex and there are so many uses for them. Initially, I was interested in them when I was working, developing products for algae barn. Basically, we had some problems to solve, increasing production for copepods, specifically our Tigriopus, and they wanted to make them redder and bigger and all that. And then there were also some bioremediation issues. And I happened, just in doing some research, I found just a little passage about these bacteria in the third volume of the Reef Aquarium by Julian Sprung and Charles DeBalt. And basically, they're photoheterotrophs, and they have been used for decades in commercial aquaculture. But specifically, they're really rich in carotenoids, which would have solved the problem of reddening up those Tigriopus pods. And then also they're heterotrophs, so they could have cleaned up all the organic matter in the cultures. And then as they grow from that, become a food for the copepods themselves. So long story short, we were doing a lot of stuff at that time. That was when Algae Barn was literally just doing pods and phyto in a garage. So they had a lot of work to do. We didn't even have macroalgae yet. So that project, the purple non-sulfur bacteria, just got pushed to the wayside. And a year or two later, when they moved down to Commerce City, which is like an industrial area outside of Denver, I opted not to move down there and instead stayed up here in nice, pretty Boulder County, or that's where I was at the time, and started. I was so interested in these bacteria by then. Once I found out a little bit of what you could do, I kept researching them and discovering they had more and more uses. And the efficacy was so well documented in aquacultural science journals and other places that I was convinced they had a place in our industry. So I had a kind of I'm a biologist by training, but not a microbiologist necessarily. So I had to kind of do a little bit of crash course in microbiology so I could learn how to locate and isolate these organisms and start playing with them. So I was literally out in a cabin in the woods for 2-3 years, up in the Rocky Mountains, with limited water resources, and
get some good thinking done there.
Yeah, so that's how Hydrospace began. We launched in 19. and it's just been pretty steady growth by then. A lot of people have been sold thousands and thousands of units by now, and by and large, it's been a very positive response.
So you mentioned algae borne. How did you actually get started in the aquarium industry?
Oh boy. So I'd been keeping aquariums for a long time. I mean, since I like to say since the middle of the Reagan administration. I've had some kind of aquarium since then. The longest I've ever been without a fish tank was when I was literally living on a fishing boat. I started as a teenager in the 80s, late 80s, at my first fish store job. So that was my foray into the industry. And I quit that job because I refused to cut my mullet. So it was a couple years before I got back in, but that was like mid 90s. And by then, the saltwater aquarium hobby had really grown. And again, thanks to the Reef Aquarium series by Sprung and DeBalk had a lot to do with that. It had a lot to do with our success in the store there. That's where I started selling corals there around like 95.
What town, city was that in?
Mandan, North Dakota.
Wow.
And if anyone's even heard of it, I'd love to see that in the comments. Yeah, I remember some of my first back when I got into saltwater fish in the 80s. I literally had to come and pick up the fish when they came in because none of the dealers in Bismarck across the river, the capital city, even had saltwater tanks to carry any marine livestock. At one point, my mom drove me all the way across, when I was a kid, all the way across the state to Fargo, just to come back with a damselfish.
That's dedication. So we got a couple of comments here. Mm-hmm. Vivid Creative Aquatics. Never cut your mullet for a job. Laugh out loud. And Jason Langer, stand up for your right to wear a mullet. There you go. So, and that we got people advocating for folks with mullets, rights for mullets there, you know. So, all right, man, we're talking right before the live stream, and it's pretty much it's like you and your wife that run this company, and you guys shot a video for me that I'm starting to play right now, and it's kind of like a tour of the facilities here. I don't know if you want to kind of talk through this, even though you probably can't see it.
Ooh, that's right. I should have been on. Well, it starts out in the kitchen. I'm just guessing if I remember the walkthrough. It starts out in what we call the kitchen with the sink, and you'll see the big kettle on the table.
Hello, ReefBum. I like
that. So that's yeah. So there's the big kettle, and that's where actually everything begins and ends in the kitchen, because that's where we brew, and then that's where we bottle. But yeah, very energy consumptive. You can actually that's a good place where we redid all the electricity. Everything's up on the ceiling, as you can see, the outlets and everything, having worked in a lot of aquaculture facilities and stuff. So kind of got to build this the way I wanted. It's all temperature controlled. That's why you get the Reflectix all over the walls and ceilings.
Gotcha.
Yeah, looks like a space age padded room or something, but pretty small, obviously. Crowded you have to buy a lot of packaging materials. I could see the wall of boxes and bottles and stuff going through, just in order to get that stuff at a reasonable per piece price. So we're real crammed down there. But by then you're probably getting into the grow room. God, I wish I could have pointed out the cubes one by one. I would have said what was in them, but you'll notice that there are a lot of different colors.
Yes.
So what you're seeing is different species at different stages of cultivation. Okay. And so they're going to have a lot of different colors because they are photosynthetic bacteria. They're loaded with carotenoids and bacterial chlorophyll. So mainly oranges and pinks, and you get some red and even like fuchsia colors, but they sit on those racks. They're just shop lights. These guys, as we were discussing earlier, are not picky about lighting.
Bacteria.
So these particular bacteria, they can adapt. So the particulars, I mean, the shop lights, I tell people how to grow it. So I'm not, there's not a lot of proprietary stuff there in that respect. But we do have them on a photo period. The reason for that is it encourages the production of these substances. It's actually the same substance that biopellets are made out of. It's basically like an energy reserve for bacteria. So when they're in the bottle, they can rely on that. It extends the shelf life, probably increases their survivability when they're at first added to an aquarium as well. But having a dark period encourages the production of that organic substance. So yeah, and then at some point you can see the kits up there too. I got a little shot of
the homegrown kits. You can
the home,
so you can grow the stuff yourself.
Yeah, so basically everything we do there can be scaled down for the home aquarist, and we do offer. It's the only thing we sell direct is the home grow kit. So that's like a bulk package for those who, similar to growing your own phytoplankton, for anybody that wants to have access to this stuff in bulk. One of those kits treats over 8,000 gallons.
Gotcha. Right, and there it is, the PNS home grow. Vivid, creative, aquatic. Some jugs look like they are under pressure, while others look somewhat deflated. Is that a result of the growth process?
Yeah, so a lot of those that look deflated, they're probably the clear ones. And those are just sitting in like slightly bleachy water. They're ready to go. You'll see a lot more of those than usual because we're pretty low inventory right now. Like I was saying to you earlier, we just got hit real hard by some of the big box companies that are already prepping for like Black Friday, and I was astounded. But yeah, we've been working so hard already for the big season. So,
Kenneth, talk to us about the main products that you guys are selling. So we see the PNS home grow. I know there's Bio, right?
Mm-hmm.
Talk to us about the different product line and what people can get from you guys in terms of the bacteria.
Sure. I would start by saying that as far as I'm concerned, these bacteria are most useful as a food. There's that bioremediation aspect, but they're really valuable as a food. They're a lot of bacteria can do the bioremediation thing, but
when you say bioremediation, are you talking about controlling nutrients for that purpose? Is that what you're talking about?
Yes.
Okay.
And in this case, because they're heterotrophs, also organic substances. So these guys can break down detritus, for example. They've been used in the pond industry as well because they are capable of digesting cellulose, which is kind of a big deal. Not a lot of bacteria possess the enzymes necessary to do that, lignocellulosic compounds. That's why cellulose is the most abundant biological material on Earth. Basically woody materials.
Okay.
Right, that plants make. Their cell wall has a cellulose. Well, as their cell wall, cellulose, to give it rigidity. Anyway, it's poorly degradable, and these guys can break that stuff down. So you'll see this in pond products to eliminate like leaf litter, stuff like that. But similarly, they can break down detritus in an aquarium. A lot of the detritus, being some of the last stuff to break down, includes stuff like the cell walls of algae, for example. So we see people using this stuff and reporting that they have to replace their filter socks less often, for example. Another thing is so if you run a refugium, a lot of those yellowing compounds similarly are difficult to digest, and these guys can do that. I've actually grown these bacteria just on tannins before, so they're really good at that. They can actually clarify your water particularly if you're running like a algae scrubber or something like that. So good at nutrients, of course. They are used in wastewater treatment for that. But they're better at removing really high levels of nutrient, and then their effectiveness tapers off as you get closer and closer to bottoming out, which I guess may be a good thing. But if you're going for like the ultra low, they're not, they help, but like I said, they're mainly useful as a food. So I like to think of them as a food that rather than polluting the water, actually can clean it.
So you're talking about a coral food, and so how do the corals? So we know that corals do consume bacteria, right? Or explain it, Kenneth, in terms of my understanding is that there's bacteria inside of corals. Let's say SPS, that there's certain bacteria inside of the corals themselves, And those are the bacteria that are consuming the food, and the corals are then getting the nutrition that way. Is that how it works?
It's really complicated. So much so
Not
that the
that simple.
no, unfortunately, but job security for me, I guess, and coral biologists. So they actually call the whole association between like a coral polyp and the zooxanthellae and all these various microorganisms. They call the whole thing like a superorganism, the coral holobiont, because they're so they almost act like a single organism. Like to start with the food, I guess they do eat, even though they incorporate these bacteria into their bodies, corals. They will eat them just like they do zooxanthellae. You know, they'll either expel or consume excess, and they're insanely nutritious. They're up to like 70% protein, always over like, you know, upper 40s, 50% protein. So, and then they don't have the cell wall like in algae, that cellulose, which is fiber.
Mm-hmm.
So they're very digestible. They have a lot of B vitamins, vitamin E, and then of course those carotenoids, which are antioxidants. So it slows the aging process, that all those free radicals. It would, by the way, that's really important in like a highlight coral, like a lot of our SPS corals, because when a coral is really photosynthesizing, the photooxidants that are produced by the zooxanthellae are actually damaging to the coral itself. It damages the coral polyp's tissues. So carotenoids, having a lot of carotenoids in their diet is really good for them. And then they say that it's hypothesized anyway in one paper that the purple nonsulfur, because they're primarily anaerobic, by the way, but they live in like areas of the gut. They find like little recesses within the coral stomach mainly, or gastro, the gut that is anaerobic, and then they kind of live there and have direct associations with the zooxanthellae, which is even more interesting and maybe more important ecologically. So coral reefs are nutrient poor environments, particularly nitrogen. They're usually nitrogen limited, not phosphorus. Corals are well adapted to nitrogen starvation. They're not well adapted to phosphorus starvation. That's where you usually see bleaching and stuff. But the reason they're adapted to nitrogen starvation is because they evolved in these environments. They've always been nitrogen starved. That's why the water's clear. When Charles Darwin first went to Tahiti, he noticed that the waters were crystal clear, and to him it looked like it was like an ecological desert, that because everything was so infertile, and he wondered why bear with me here, this is really interesting, why, and really important. He wondered why the water was so clear, and yet the coral reefs there were so productive. This environment was just, you know, rich. And that's one of the few things he wasn't able to figure out in his lifetime. And ever since that, biologists termed that Darwin's paradox. And in the 70s, this expedition, Symbios, I think it was called, discovered the reason why corals can be so productive in these nitrogen-limited environments. And it's because of nitrogen-fixing bacteria. Nitrogen-fixing bacteria basically do the reverse of denitrification. What they do is they take nitrogen gas and they convert it into ammonia. So that's how new nitrogen, bioavailable nitrogen, is introduced into an ecosystem. And these bacteria are very competitive on a coral reef because it's a nitrogen-limited environment. They're able to compete really well with other microbes. And indeed, they live inside of the corals and have these direct associations with the zooxanthellae. So jump back into that. Basically what like Rhodopseudomonas does, that Rhodopseudomonas palustris, the bacteria that's in actually all of our products, but PNS ProBio is just Rhodopseudomonas palustris. That species is found in corals, and the abundance of that species within corals changes seasonally in some cases, depending on nitrogen availability, believe it or not. But what they do basically is, you know, algae, there are no algae, only certain bacteria can fix nitrogen, and no algae or anything like that. So they're very important. Basically, they provide nitrogen, bioavailable nitrogen to the zooxanthellae, and then the zooxanthellae can be more productive, and that benefits the coral. And then, of course, these bacteria feed off the coral's wastes.
Gotcha. A very complicated process going on there. I guess one thing that I hear in terms of skepticism, in terms of dosing bacteria on a regular basis to an established reef tank is, why do you need to do that? I know when I was talking to you about, you had sent me some stuff for my systems, and so I had a bunch of questions in terms of the dosing regime, and my understanding is that when you're dosing a bacteria, whether it's your stuff or another company's bacteria, that it should be done on a regular basis. And I think the skeptics that I've talked to, including on the show, have said, why is there a need to dose bacteria? Because bacteria can multiply so quickly once they're added to the tank. So yeah, go ahead, man. Debunk that theory in terms of why you don't have to dose bacteria on a regular basis.
Yeah. Now, I can't speak for other companies or, the bacteria they make. I can't even speak for companies that make the same type of bacteria that I make, if there are any or will be any, because even those will be different products because of the culture media that they use and all that. And even the strains. Our bacteria, so Rhodopseudomonas, the question shouldn't be, will it live in my tank or not? The question should be, will my tank benefit from this or not? Right? So if it doesn't survive, that doesn't make it not beneficial. When we add calcium to our tanks, it gets depleted. So does that mean, oh, well, what's the point of adding calcium to our tanks? Right? It's a weird line of thinking. So the question should be, should these bacteria be in my tank if I want the microbiome to look like a natural microbiome? Researchers down in Australia they've been really ahead in this field. I think James Cook University, there's a paper by Professor Jessica Tout. They look at different niches within a coral reef, a healthy coral reef in the Great Barrier Reef, Heron Island Reef, I believe, and Acropora, can't remember which species, but it's Acropora they were looking at specifically. They were looking at the mucus, the water column, the sediments everywhere. And in all of those, purple bacteria generally show up a lot. And there's only like the top 18 species, genera. And there might be hundreds or even thousands of genera on a coral reef of bacteria, right? These guys are in the top, multiple purple bacteria species are in the genera, or in the top 18. That would include Rhodopseudomonas, Rhodospirillum, and Rhodobacter, all of which we have in our products. So it's like, do you want your microbiome to look like a natural reef so it functions ecologically like a natural reef? If you do, then you want to have these bacteria that in there. Now, it may be true. Dr. Tim pointed this out on your show before. He's looked at some tests from Aquabiomics before that actually suggest that purple non-sulfur bacteria tend not to survive. They tend not to persist in the typical aquarium. And I believe that's correct.
Why don't they persist? Like, some bacteria do multiply rapidly, is my understanding. But why would that particular bacteria not be sustainable in a reef aquarium?
One would be maybe not the main reason, but one reason would be that we don't have relative to a natural coral reef, we don't have enough anaerobic microhabitat, especially nowadays that people don't like deep sand beds as much as we once did. So these bare bottom tanks aren't really hospitable for anaerobic bacteria. And then the other component would be competition with other bacteria. So these guys can live in an aerobic environment, though not photosynthetically.
Can you just quickly explain aerobic versus anaerobic in terms of the difference between the two, just for us non-scientists?
Yeah, so an aerobic environment is obviously just one that where there's some oxygen present. Anaerobic is where it's lacking. Earth, at 3.5 billion years ago, Earth was completely anaerobic when these organisms evolved. And they had a lot of the descendant of these bacteria, actually, these photosynthetic bacteria, is cyanobacteria. It was cyanobacteria that changed the Earth's atmosphere to make it oxygenated, right? Because they were the first oxygenic phototropes, the first photosynthetic organisms that make where oxygen is a byproduct of photosynthesis. So there was a great extinction event, actually, and a lot of those microbes disappeared, but these guys survived because they were able to adapt to an aerobic environment. Oxygen is still poisonous to a lot of microbes that are obligate anaerobes. So there you have your obligate anaerobes that have to live in an anaerobic environment, and then you have your facultative anaerobes that can live in both, but they prefer to live where there is no oxygen. And indeed, these guys actually they can swim, and they'll swim away from oxygen to move to a less oxygenated environment.
So let's get back in terms of the benefit in terms of providing food for the corals. So what is a how do we know that our corals need, certain bacteria to be healthy and to thrive? I mean, is it something that can be tested via the aquabiomics to kind of see if we're deficient in certain, bacteria? Is it the general look of a coral itself in terms of whether or not it's not thriving, not growing? How do we know that the addition of bacteria on a consistent basis will be beneficial to our corals? I guess both visually and potentially with data.
Yeah, and it may not be. It depends on the situation, right? It depends on the actual aquarium system. As Dr. Meyer pointed out in your show of aquabiomics, he's noted that there is a lack of Pelagibacter in the aquariums. That's the most abundant marine bacterium on Earth, and corals feed on that species or that group. So say you run a UV sterilizer, you're killing a lot of those, as he pointed out. So you're depriving your corals of a natural food source, an important one. So much so that corals actually produce substances in their mucus to bait them. Just really quickly too, if anyone's wondering, that's how corals eat bacteria, is they capture it in their mucus. And bacterioplankton actually form the bulk of most corals' diet, by the way. It's not phytoplankton. Phytoplankton, I saw in one paper, was like they figured 0.3 to 13% of a coral's diet is actually phytoplankton. And a similarly small percentage is devoted to zooplankton, and the rest of it is bacterioplankton.
So I've been on and off with UV on my systems, and I have SPS dominated tanks. So I've for the longest time never ran UV, and I did really well, in terms of growing corals. And then for a long stretch I ran UV, everything was doing great. Then I stopped running UV. It was kind of hard to tell the difference between using UV and not using UV in terms of the benefits to the corals in my tanks over the years. And others that run UV report having great looking tanks in terms of the animals in their tanks. How do you get to that in terms of, because I've run a lot of data with Eli and aquabiomics, and so I have seen what you're talking about in terms of that particular bacteria not being present in my system. So I guess my long-winded question to you is, given what you just said in terms of that bacteria and the importance of that bacteria and UV, is it critical to, or would you recommend running a reef tank without a UV sterilizer for that reason?
Generally, yes. I don't see necessarily that they would cause a lot of harm. I've seen a lot of awesome tanks. I still run UV, like I have like a goldfish, it's a fancy goldfish collection, I use UV. It has its uses, quarantine, places like that, but on like a mature reef tank, I'd prefer not to have it. It just kills too much of the microplankton. It's one of those, it's a preference, but going back to why is a bacterial product good, or, you know, if I have to keep using it? Well, that's part of the reason you have to keep adding these things. Planktonic bacteria are different than benthic bacteria. Sometimes there's some overlap depending on, you know, like some have multiple life stages and so forth. But if you're killing all your bacteria in the water column, then you're depriving your corals of a natural food source. And there are other implications in like nutrient cycling and things like that that are equally important. It disrupts the system completely. Whether it ends up being good or bad overall just depends on the specific situation.
Are there any telltale signs, you know, in terms of physically, you know, looking, observing your corals, that you could tell that they're missing that benefit of having certain bacteria in the system? Like, would they be paler or less polyp extension, those sorts of signs?
So it's one of those things where you wouldn't necessarily know, because you wouldn't know it happened if you hadn't used it, if that makes sense. Mm-hmm. For example, when it's benefits as a probiotic, say it may have prevented a vibrio infection that you would never know that if you hadn't used it, your whole tank would have got wiped out by STN, or RTN. So it's kind of hard to say. I wish there was more literature about these bacteria applied to corals in captive systems. There just isn't yet. It's all implication based on everything I draw from is implication based on observation of wild ecosystems. So it's all anecdotal, but there's a lot of it now, right? If you go on Reef to Reef for years now, there's just that one place, there are hundreds of people that have used this stuff and reported, their experiences there. Just last night, I was at an LFS. I didn't even know they carried my products. And I was talking to one of the guys there, and he said he uses it on his corals, and he noticed he was talking to somebody who was using it. He said he uses it and likes it, but someone else was using it, and he said that they had their corals same place under radions, and didn't do anything to them until they started feeding these bacteria to their corals, and then all of a sudden they started to color up. And I didn't ask him specifically about coloring up, what colors or how much or anything like that, but he said they colored up. And that could be, for example, because of the carotenoids. I mean, it's all anecdotal, right? You can't, it's impossible to say, but mainly the health benefits are just as like a food. Like I said, it's a live food, it's a natural live food, and it's one that actually acts as a probiotic in the sense that it inhibits disease organisms.
So Greg Carroll had a question. You mentioned some percentages before, and I think the question Greg is asking is, are those percentages based on the amount available of those foods to the coral and the reef with so many filter feeders abound? So I guess there's variables in play, right?
Tons, yeah. So in that particular paper, that was their estimate. It's just a number that came to mind, 0.3 to 13%. And which is obviously, that's a pretty wide window, but I mean, 13% being the highest estimate from that one researcher's perspective, 13% isn't all that much. By the way, that's not to say that phytoplankton isn't important or beneficial in a coral's diet. It's one of those things where a small amount can go a long way because of the vitamins and especially fatty acids that are available in algae. Tross will be talking to you more about that later this month, I think. But yeah, a little goes a long way there. There's actually a study that looks at these bacteria, Rhodopseudomonas, the one that's in PNS ProBio. PNS ProBio, by the way, is my product placement. These bacteria, by the way, the red color, these are the carotenoids mainly. So you can actually see it in there. That's how you know it's not just a bottle of RO water or something. Very distinctive carotenoids. But there's a study, they used brine shrimp as a model organism, but it could be anything because we all need DHA and all of these certain fatty acids that animals can't produce themselves. They have to get it in their diet. But anyway, they used brine shrimp as a model organism, and they found that using Rhodopseudomonas compared to phytoplankton, one wasn't necessarily better than the other. They were best used together because they're complementary. Again, because the bacteria is really high in protein, has the B vitamins, whereas the phyto has certain fatty acids that are really beneficial and other vitamins. So yeah, they're better used in tandem. Algae Barn knows that. That's why I think Algae Barn has a lot of. They sell like in packages these bacteria with their phytoplankton together in a lot of different like bulk. I'm sorry, like package deal things.
Gotcha. So you, Kenneth, you mentioned probiotic before, and I see Mark Clark says comment about helps keep the coral bacteria. I'm assuming it meant bacteria keeps the coral's immune system strong. And yeah, you talked about bacteria as a probiotic and being able to kind of potentially cause a coral to fend off pathogens. Can you talk more about that? Yeah,
the big one would be Vibrio. Vibrio is well known in our industry due to its ability to attack corals, especially a Vibrio coralliticus, and as in the wild. And these bacteria are the natural enemy of Vibrio. They have very similar niches, with these guys having the benefit of being photosynthetic, maybe. But the big difference is that whereas these bacteria are always beneficial to the coral, Vibrio is an opportunistic pathogen. So Vibrio actually benefits corals in different ways. Actually, in the wild, it's usually just sitting there latent. It's everywhere. I've done aquabiomics tests of the Reef Builders studio, one of their systems, and it was rife with Vibrio. It's always sitting there, but it waits for a coral to become compromised in some way, and then it attacks. What's really interesting is that the Vibrio know to attack because when the coral, especially certain types of stress, will disrupt the interaction between the coral and the zooxanthellae, and the result of that is a lot of DMSP production, which is it's a substance that reduces oxidative stress, osmotic stress, I'm sorry. And the Vibrio has evolved to sense that, and they're actually chemotactic to it, meaning that they swim towards sources of DMSP. So that's how they locate stressed corals. And then that actually induces changes in the Vibrio so that they become, it makes them produce virulence factors. It increases their swimming speed. It does all kinds of weird stuff that turns them into all of a sudden they just, turn into Brutuses on the coral, right? Now, where do these corals come into play? Well, they compete with Vibrio for space. They also produce antibiotics such as streptomycin, which are known to have activity against Vibrio. In fact, these bacteria have been widely used in the Pacific white shrimp industry, specifically to combat a particularly nasty type of vibriotic disease in those shrimp. So used as a probiotic in farms and stuff already with a well-known efficacy. But in the case of corals, these bacteria are already resident on the coral, so that they just kind of fight off the Vibrio in that way. And lastly, they actually metabolize DMSP. So they can actually disrupt that signal that goes out to the Vibrio telling them that this coral is stressed. So the Vibrio isn't made aware that the coral is stressed, and that buys time for that coral to actually recover before it's inundated with these now virulent Vibrio cells. One example.
So circling back, Greg Carroll says he's asking that question about the percentages because it could be argued that if you feed more phyto, they could benefit even more. Just like we know higher alk can drive higher growth. Is there a balance in terms of dosing bacteria and feeding live phyto? What I guess what are the benefits of doing both? I mean, obviously they're both foods for corals.
Yeah, they're nutritional benefits. So, we can't live off of bread alone. That we have to eat a varied diet so that we get all the stuff that we need, and perhaps not too much of some things. But in nature, if we're trying to mimic nature, 5-10% of corals' diet really should be phytoplankton. At that point, they're getting all of the, fatty acids they need, for example, and things like that. There's no harm in overdoing phytoplankton, just as there isn't any harm in overdoing these bacteria. We've done 40 time overdoses on test tanks with SPS, LPS, zoanthids, softies, and 40 time overdoses. So that's a whole bottle of Probio in a 10-gallon tank. And the corals, it didn't hurt anything. The corals actually just opened up like they were really feasting.
Is there any advantage to actually feeding live phyto versus non-live phyto products?
I know people argue that. I've used both. I've used a lot of paste in hatcheries before, and it's convenient. I mean, that's the only reason it exists, is because it's convenient. It doesn't exist because it's more nutritious. Definitely not. But I like live phyto. It's just cleaner. Obviously, and even if you just freeze something live, you're going to lose some nutritional value. Not much, but anytime something isn't live and living, it's not as good as it was before. Remember, some algae actually are motile and they'll swim around, so they'll stay in the water column longer, and they're doing things and secreting things that are telling the corals are selective filter feeders. So that, they're sending out these cues that they're nutritious and palatable and yummy. So that's, I think more corals end up eating it, and it's actually more nutritious that way, and it's certainly cleaner.
So Sonny from Reef Site is asking a question, and I had him on the show. He's a big advocate of dosing bacteria. He says, do we really need phyto? I have a very successful reef, and I've never dosed it in my current system. My system relies heavily on bacteria. So a lot of people don't dose phyto and have good-looking tanks. Is that something that you mentioned? It's a small percentage, I guess, in terms of what the corals would consume. So I guess to answer that question,
small but important. I mean, how much vitamin C by weight do we eat in a day? But we'll die without it horrifically. So would I use phyto? Yeah, every day personally. But it depends on what's most important is that the corals are getting the things they need to live in their diet. So it depends on what else you're feeding them. There are a lot of artificial foods now that include those things, but I would argue that live foods are by far they're not the most convenient thing, and they're not the cheapest, but it's hard to argue that they're not ideal in terms of nutrition and also palatability, which matters, especially to selective filter feeders.
I want to go back to a question that somebody asked earlier in the chat. I think it's an interesting question. Thomas the Cat. I would like to know the effects of ChemiClean and peroxide on the bacteria in the tank. I'm almost certain they kill back bacteria on a mass scale. So what are your thoughts, Kenneth, in terms of using something to knock out cyano like ChemiClean? And a lot of folks use it. It's got its pluses and its minuses, but are those minuses something that will catch up to you later?
It could. I think it's a erythromycin, right?
Yeah.
Like gram positive. You're wiping out like a potentially huge. I mean, it's extremely disruptive to your microbiome. So, I mean, you're throwing dice. I mean, we don't really know what I suppose in extreme cases, maybe it's like as a last ditch effort to use something like that. I can definitely like see service company techs wanting to, wanting like a quick fix. They don't want to be joking around when they have a client that, especially like at a business or something. But I don't like anything like that. Ozone, anything that's going to kill the microbiota. It's usually just hiding the problem anyway, and not actually fixing it.
It's a band-aid on the problem. I guess something that some things that people do when they dose something like ChemiClean is that they will dose a crap load of bacteria to try to replenish that bacteria that potentially can get wiped out by that type of product.
Sure. We don't really know what we're removing,
theory,
so
right?
what should we know to add back? I mean, it's kind of hard to do. So, like people have used this, my products specifically after treatments like that to replenish beneficial bacteria in their aquarium. And I'm like, okay, that's cool because you're adding something beneficial. But if you wipe out, hundreds of species, you can't add all this back without, maybe some live rock, live sand. There's a different, that actually is an interesting, so people often, there's this debate, should I use bottled bacteria or live rock? And when discussing biodiversity, and they're just two completely different things, right? So with different purposes. So live rock, live sand, those things are good for adding diversity, obviously. Because they're going to have a lot of species on them. Some of them bad maybe, but if you're just going for diversity, that's the way to do it. The reason to add a bottled bacteria is that you can add in concentration a known beneficial bacterium, and you can apply it for a specific purpose. So say you have really high phosphates and you want a species that's a phosphate accumulator. In other words, a species that can take up more phosphate than it needs for its own metabolism. These bacteria do that. They can be 15% of their dry weight can be phosphate, because they'll like take it up and store it for later use. And then of course they get skimmed out, and you export tremendous amounts of phosphate that way. Cool. And you use a species that can do that. That's where bottled bacteria have their advantages. Like if you want to use them for a specific purpose, or a food, as in the case of PNS ProBio.
You mentioned skimming, and we talked about UV. If you're dosing, your products on a daily basis, how are you supposed to handle the skimmer and the UV? Should that stuff be turned off for like an hour after dosing? Should it be turned off for a longer period of time? That could get difficult if you're using UV 24/7.
Mm-hmm. So I wouldn't use UV, but some people do. And if they do, and you're just using this as a food, don't worry about it. So if you irradiate these bacteria, they're still flowing through the system. The corals will eat them, and it doesn't really matter. Right?
So what percentage do you think would get irradiated by the UV?
Well, that would depend on the turnover rate of your UV unit, right? But and then how fast it takes for the corals to eat the, I guess like if you ever overdose phyto in your tank, something that's really, that's conspicuous green color, and some people see that clear up in an hour. It just depends. If you're using it, so say you're trying to seed the tank, like with these bacteria, with the hope that they'll live in, say, like a anaerobic biomedium or deep sand bed or something. I saw one study that saw, so Rhodopseudomonas was immobilized, in other words, attached to a substrate from its free swimming stage within 60 minutes. So it depends on the turnover rate, and then how much of the UV unit, and then how much you're willing to lose. So if you look at that and you're like, oh, I may lose half in an hour, I might just do a double dose then, rather than turn off the UV, right?
So go ahead.
I was just saying, some people may not don't like turning off their skimmer off and on, because it's inconvenient, or they might have a pump that jams, gets irritated, won't turn back on, or something like that. But there are benefits to that anyway. Like if you just run it at night, for you have the added benefit of pH stabilization, I guess. But
Right.
you know, like Dr. Tim, I think again on your show, recommended doing that, just having like a intermittent, like a skimming period each day, out of each day, like just running it at night or whatever, and then that would preserve some of the bacterioplankton, because they reproduce very quickly. You know, some of these they'll become great grandparents within 24 hours, right? So that gives them enough time to reproduce during that off period where they're, you know, to maintain some kind of population.
Would you say it's a safe bet to, if you're going to be dosing bacteria every day, to be turning your skimmer and UV off for like an hour after you're dosing? Is that a good rule of thumb, or would you do it longer?
I just leave them on. I think, and again, I keep referring to past guests on your show, but Sanjay was on talking about
he's a big skeptic of
study.
dosing bacteria.
Yeah, and he's bought my stuff, but he did a study that seemed to indicate that skimmers do remove massive amounts of bacteria, but when you press him about it, he'll say, "Well, I'm not really sure." You know, it is not sure is or how, you know. So you know, how much it may depend on a lot of things. Certainly it depends on the skimmer, the efficiency of the skimmer, and the way you and the settings and all that. But if you're removing some, I don't see where it's worth turning it off and on. It makes things harder. These products, I'm not going to say they're cheap, but they're not that expensive. If you throw it in and you remove a little bit of bacteria come out, I mean, that's totally fine. In fact, if you're using it for bioremediation, eventually anyway, you want some of those bacteria to be removed by your skimmer anyway, because that's how the stuff they sequester gets exported in the first place, right? So again, I would just run it on like a day and night thing if that were possible. Otherwise, I just add a little extra product and call it good.
Gotcha. So let's talk, Kenneth, a little bit about the different products that you have and the different use cases. So PNS substrate sauce. When's typically the best time to use that in the life cycle of a reef tank?
Certainly at the beginning. So that is when we first came out with PNS ProBio, people were asking, they wanted to use this for cycling.
Right.
And it's not a nitrifier, but it does complement nitrifying bacteria in multiple ways, specifically because they inhabit different niches. Most notably, nitrifiers are highly aerobic, hence like a wet-dry filter and things like that. These, of course, they prefer an anaerobic environment. So they live in two totally different areas. And then these guys are heterotrophs, so unlike nitrifying bacteria, these guys are able to consume organic matter, organic wastes. So if you're doing fishless cycling, that's not too important, but as soon as you add fish, it will be, right? Then you have something there in place ready to consume all the fish poop and other wastes that are generated once you have all that life in there living. And then lastly, whereas nitrifying bacteria tend to produce nitrate, Rhodosudomonas anyway, one of the two species that is in substrate sauce, this product actually has two species of purple non-sulfur bacteria in it. Why? Because it came out after PNS ProBio, and I had a second species by then, really no other reason. But I would have put both of these in both products, but it does make PNS ProBio cheaper, which helps keep the cost down, like if you're using it for a food. So again, that one's your general use, the one you'd add all the time. But this one is mainly just for cycling, substrate sauce. One thing about substrate sauce is that it actually we put an excess of phosphorus in it. So phosphate, dipotassium phosphate. So that's because nowadays with fish, fishless cycling, with like dry rock and stuff like that, we actually don't have enough phosphorus in the system to promote the prolific growth of microbes that we want during the cycling period. So it actually has some supplemental phosphorus in it. I point that out because I wouldn't want people using that in their regular tank if they already have high phosphates.
Now
I do know there are some people that use it for that purpose. It really wasn't intended originally to be used for that for people that are dosing phosphates.
Yeah.
It's a cycling product, but there's no reason you couldn't use it for anything really, other than that excess phosphate. And some people now, I guess, that are dosing phosphate may actually prefer to use substrate sauce because it does have that supplemental phosphate in it. The last thing about substrate sauce, though, comparing it to PNS ProBio, is substrate sauce, the cycling product. This is actually cultivated in salt water. So this is the only product that's saltwater specific. And the reason we cultivate it in salt water is so that it acts more quickly when it's added to a system. So it shaves about 3 days off the lag period, which is basically that period where bacteria are adapting to their new environment and kind of waking up, so to speak. Since people generally want to cycle their tanks as quickly as possible.
Yep.
We just made something that was saltwater specific so that it would you put it in the tank and it just starts going.
So, all right, now you mentioned PNS ProBio, and that's something more so for an established reef tank, and that's really all the benefits that you were talking about before in terms of being a probiotic, providing nutrition for the corals, and also helping with nutrient control. Correct. So let's talk about the frequency of dosing, and I think my understanding is that daily would be best. I mean, you did mention that it can get pricey. Right, if you have a larger system, you need to do daily. If you're on a more of a budget and you can't afford to do daily, what would be the next best thing? Every other day?
Well, can you afford massive water changes and pallets of lanthanum, chloride and all the other. So the idea is that this is a live food. It's more nutritious, so you get more out of the food you add. More growth. It's a probiotic, so it eliminates, it'll actually reduce the incidence of disease, and instead of polluting the water, it cleans it. So you think of the price of salt or RO water, all those things combined, it becomes more clear why these are, Asian businesses are not known for spending money like drunken sailors, right? They use this stuff widely in their hatcheries. Why? Because there's a humongous cost. There's advantages from a cost benefit perspective overall. So we actually have some clients that have massive tanks and some coral farms as well. But they found out very quickly that as a bioremediator, just in the terms of savings of time. If you're say you have like a store or an aquaculture facility or something like that, where in addition to the material expenditures, you have wages to pay, and all those other things consume labor. So you factor all that in, all of a sudden this stuff doesn't look so expensive. Now, if you're home hobbyist, it depends. It scales differently, but that's why we have like that PNS grow kit. So if you, the PNS home grow, so if you're a home aquarist, that 8,000 gallon treatment, even if you have a 200-300 gallon aquarium and you're feeding every day, that lasts pretty well. And economically, it looks pretty good in the end, all things considered.
So talk to us about that. I was asking you some questions before the live stream in terms of you need to keep things sterile. You got to have sterilized equipment and all that stuff. What does that mean in terms of sterilized equipment? Are you boiling instruments and stuff like that to make sure that, or using hydrogen peroxide to make sure that things are not going to get contaminated? While you're culturing this bacteria.
Depends on the instrument. So. You can so we use heat for the culture medium itself. You boil it to sterilize that. And basically you just have a packet of dry stuff that you add to boiling water.
So that's the sterilization. You're just boiling the water.
Well, the water that will be the culture medium.
Right.
Yeah. And so that is hot. And then you add it to the culture vessel, which is bleached prior. So there's bleach.
That comes bleached.
No, you just add like a little bit of bleach and shake it up and then pour it out. Then we actually don't recommend using tap water to rinse that bleach water out, because so there's like you add like a cap of bleach. This doesn't have to be exact, but you put about like a cap of bleach in a cup of water and shake it up in there. Just make sure you get all the inner surfaces of the culture vessel, which, by the way, is like it's a clear collapsible cube. So you just shake it up in there, so you just shake it up in there and sterilize the inner surface, pour that out, and just the last few drips, you shake them out, because we don't rinse that out. Your tap water lines have some pretty resilient bugs in there that are spore forming and get in and contaminate stuff. So that little bit of bleach doesn't do any harm. And then the other, like anything you stir with, or like your funnel and things like that you may use, we suggest peroxide for that, because you can just put that in a mister and spray it all over then, and then that's
and so you can't
pretty effective.
You can't reuse the home grow kits, right? You basically have one home grow kit, and then you got to move on after you culture the bacteria to get to another kit, correct?
Yeah. So the reason for that is that heating the cube actually breaks it down pretty badly. So repeated heating like that isn't good for it, and I wouldn't want someone to burn themselves. That's still 2.5 gallons of boiling water that just, and then, but the main reason, other than safety, which is a good enough reason, is that by far the most money and labor goes into producing the culture medium itself. So that little packet of powder contains a whole bunch of different ingredients, and every single packet for each kit every single ingredient is measured to the 100th of a gram individually. And that way we don't have settling like we see in our sea salt buckets and stuff like that. It just ensures really consistent results. So things like trace elements or whatever, double or tripling or not having it there can make the difference between your culture turning out or not. So basically that's what makes, I remember Jake Adams used to give me crap about that. It's like, I just can't understand why it's so expensive. He's like, it's like these ingredients are just like a buck a piece or whatever. I'm like, yeah, but you have to order them all individually. You measure them, and it's like, and then he kind of got it, but it's easy to see that from the outside and go, well, it wouldn't cost that much to put the kit together, but it does in terms of time. If you want them to work optimally, they're guaranteed. We'll replace it. I've only had one kit that ever out in the wilderness that didn't turn out, and that's because she tried to put it outside on her porch, so the temperature and the lighting both fluctuated wildly. Yeah. I replaced that kit, and that one grew perfectly well. So basically what I want is for people to succeed with those. You grow so much, it's like on a like a per milliliter basis, it's pretty cheap anyway. So I'd rather focus on success than cost.
Greg Carroll's asking, can you microwave it to sterilize it? Is that a possibility versus boiling?
I guess you could. Yeah. I mean, if you have a way to like microwave 2.5 gallons at once. If you have a giant microwave, I guess.
Depends on the microwave oven itself. What was I going to ask? So you mentioned temperature and whatnot. Is there a certain temperature that you should be storing this stuff at?
So these guys are thermophilic. They actually like really warm temperatures. The shelf, like you can keep them on the shelf at like 112, 114 degrees indefinitely. A lot of my tests are kind of like accidents that happen. One time I accidentally left a bunch in the back of my Jeep in the summer, and I looked up a heat index thing based on the temperature of that day or those days, and it was like over 130 degrees for extended periods of time. Whoa. So I used those bottles to try to make new cultures, and yeah, they grew like nothing had happened to them. So didn't really matter. So as far as shipping and stuff like that in the summer, I wouldn't worry about it. And you can certainly, this is the question most people would be, you can certainly store them at room temp. In fact, that's what I'd recommend.
What about the other end of the temperature spectrum? What about shipping in really cold temperatures? Should you not do that?
Yeah. So Rhodopseudomonas, anyway, the strain that we're about to introduce, the new strain, that one I have tested for freezing. And it actually survives, it remains viable after freezing and thawing. And again, I used it to cultivate and to make new cultures, and it was almost like nothing happened to it after it had thawed out. So again, yeah, shipping in the winter, it can, you can come home in January from work and it's literally like an ice cube on your doorstep, and
Be fine.
just thaw it out. And
resilient buggers. What about
yeah.
What about, how do you know a culture has crashed?
So one cool thing about these bacteria is that they're so conspicuous to the naked eye, right? So you can see that they're alive because the carotenoids, as soon as they die, they just start breaking down.
So the reddish color is an indication that that thing is they're what color would it turn if they did not survive?
Well, it just kind of start going clear.
Okay.
But if there was a contamination, you might see other colors in there, like especially like black, if you had like sulfate reducing bacterium in there. And then of course the smell would change too. It's not, now these products don't smell, they're anaerobic, they don't have a pleasant smell as it is. ProBio has like kind of a weird kombucha fermented smell. But which doesn't, it dissipates as soon as you open it to the air, put it in a tank or whatever. But if it spoils, you're going to smell that more like a spoiled food smell.
Lovely.
Death smell, for lack of a better word.
So Sal Spinato made a comment that having used the ProBio for a couple of years now, I can vouch for the quality of this product and how amazing it is at eating detritus. And so I wanted to kind of follow up on that comment and ask you about, you mentioned cyanobacteria. Is ProBio potentially something that can be used to help prevent cyano or help eliminate cyano because of the effect it has on detritus?
Yeah, I would say potentially. I haven't seen any interesting. I saw a study where they use these bacteria like in a larger water body to clean up after a cyano bloom, to clean up everything that was left after it was dead. But I don't know. So it makes sense though, because the cyano are actually still, they're also susceptible to the antibiotics that these guys produce, the streptomycin, kanamycin, and they have similar niches, right? They're both phototrophs. So they're both going to be competing not just for space, but specifically for space where they can capture light. And cyanobacteria are also heterotrophs. So they will also compete for organic matter. And that's kind of the most important part of that, really. So like in a sand bed or something where you tend to see cyano growing right at the top of a sand bed, they're capturing all the dissolved organics that are coming up from the sand bed. If you have, now these guys would live somewhat below in the sand bed, right? Because they like a more anaerobic environment, but they would theoretically intercept a lot of that upflow, if you will, of organic matter and limit the amount of food that the cyano get. In fact, I don't know if you know what a Winogradsky column is, but a lot of us may remember in like high school biology class, you have these tall, clear columns and you pack them with mud and then put them in the light in a window or something, you cover them, and then you'd see all the different colors form. And what you'd see on the top is like something, it would be green, or so you'd have like algae or cyanobacteria on the top. And then right below that, you start seeing like a purpley red, which is purple non-sulfur bacteria, such as these. And then you might get purple sulfur bacteria underneath that. Those guys are obligate anaerobes, so they live deeper down. And what they do is they actually eat the sulfur that, now I'm going off subject a little bit, but this is cool. They eat the sulfur that comes up from deeper down, that black layer, the sulfate reducing bacteria. And we're actually cultivating some of those that I harvested from the wild. So that's a future product we get a chance to mention later. But anyway, I went way off subject there. I can't remember what we were, the original.
I think the my original question was the use of the products to help prevent or clean up cyano.
Oh, right, right. Yeah. So because of where they lie in the substrate, they will actually consume a lot of that organic matter, and thus compete with cyanobacteria for food.
What about
I also believe.
Go ahead.
It's just and also the antibiotics potentially could inhibit cyano.
What about the constant use of activated carbon? You know, that stuff helps remove organics in the water column. What are your thoughts in terms of using activated carbon 24/7 on a reef tank? Is it good, bad, indifferent about it?
Both.
Yeah.
Both, I guess. I mean, if you have some people, they'll say it removes good stuff as well. So I think overall it's good. I like using it. I use it all the time. The one cool, you can actually use that stuff with these bacteria. I've seen some stuff in aquaculture and in soil stuff too. I've seen people, because biochar, right? Aquachar is, I guess, is how we may be familiar with biochar in the aquarium industry. But biochar is a certain type of activated carbon. And anyway, these bacteria were shown to really like growing on biochar for some weird reason. And I think they like the porosity. So the carbon will actually attract a lot of organic substances, but the matrix of the chunk of carbon makes a really ideal habitat for these bacteria in the first place. And then once they walk in, there's already a food source. So yeah, it was like one paper, it was like a soil science thing, but these rhizobial bacteria, the general group that Rhodosudomonas belongs to, and some types of bacteria actually went down on biochar, but these guys, their populations increased by about 11%. Compared to, it was like Hagen Biomax or some kind of sintered glass medium. So yeah, carbon is you can use it for both, or you can use them together actually. But as I mentioned, one cool thing about these bacteria compared to a lot of other heterotrophs is that they consume yellowing compounds. So if you have an algae scrubber or something like that, you may not even need as much carbon. So these bacteria may actually reduce your need for activated carbon in some instances.
What about carbon dosing with your products? You know, carbon dosing can accelerate the bacteria in a system in terms of having it proliferate at a very rapid rate. It can be quite dangerous depending on how it's utilized. But is that something that people can do with your products in terms of carbon dosing to kind of help the bacteria multiply even more?
Yeah, depends on the carbon source. They don't really like alcohol, so ethanol wouldn't do much. Vinegar, on the other hand, they love acetate, so vinegar would be a great one. The only issue, if you're using our bacteria, you kind of eliminate the need to carbon dose, right? The reason we carbon dose in the first place is to increase the population of bacteria so that they consume more of the nitrate and phosphate. But if you just put the bacteria in there, you can see that reduction of nitrate and phosphate without the added risk of a massive bloom of aerobic bacteria, right? So when I worked in stores, we sold a lot of no pox, no PO, however you want to call that. And a lot of people had die-offs in their tank because the mindset is, well, I have a lot of nitrates or a lot of phosphates, so I'm going to use a lot of this stuff. And then they have a massive bloom, and then it's mostly aerobic bacteria that consume it. And they can, all of a sudden the fish are gasping at the top and corals are closed up. So that stuff can be kind of risky. And it's like if you're dosing bacteria, you eliminate the need to do that in the first place. Rather than trying to grow those bacteria, you just put them in there and they'll do their job. In so doing, these bacteria, because of their ability to degrade cellulose and other recalcitrant organic wastes. These guys will, instead of like the vinegar or vodka or whatever, they'll actually eat your freaking detritus. Right? So do you want to add a bunch of, I just drink the vodka and let them eat detritus, right?
That's a good way to do it. Chris from ACI, Chris Meckley is asking, what do you know about the types of bacteria that utilize UV or infrared photons proliferate?
UV? I don't know. So these guys can use near UV. I don't know of any bacteria that, like for photosynthesis anyway, I don't know of any bacteria that can utilize UV. But I know he's looked into that. Hey, Chris, I know he's looked into that a lot. Some kind of mysterious benefits of measured uses of UV. I can't really speak to that, like with anything I know about these bacteria, but I assume they don't like it just because of the sorts of environments that they live in. They usually aren't exposed to a lot of UV.
Hey, Rich Colombo. Back to the carbon source, but won't a carbon source grow their bacteria strains that might be in the tank?
Yeah, so if you're carbon dosing, you are you're promoting the growth of all kinds of heterotrophs, right? And some of them may be bad. That's just another potential disadvantage to carbon dosing, is you may actually be promoting the growth of a disease organism. So, if you're using a bottled bacterium, you don't have to experience, say, like the drop in pH and all the other drop in a DO, all the other bad things associated with carbon dosing. You just add the bacteria rather than trying to grow it in the tank. It's safer, and what you're putting in there. You're not going to be growing a bunch of Vibrio or something like that.
Right. What was I going to ask you? All right, so you'd sent me a bunch of pictures, Kenneth, of some different bacteria. Did you want to run through that stuff?
Yeah, that'd be cool. I think I remember the order, thankfully, because I don't see them on the screen. But
I got the. So you gave me like 9 different ones. There is no freaking way I'm gonna be able to pronounce these, so I got to say number 1.
Yeah. So the first one is Rhodopseudomonas palustris. That is rod shaped, or you may say hot dog shaped, and that for that guy. The individual cell, they vary depending on the cell shape varies depending on environmental conditions, but that's like the individual Rhodopseudomonas, couple microns long, so really tiny. And the number 2.
So what's the significance of that one?
Well, that's the one that, again, that one is mostly when I've been talking about food, that one is an excellent food. Okay. Yeah, it actually also lives inside of corals in natural coral reefs and is the nitrogen fixer.
Okay. All right.
So it provides nitrogen to the zooxanthellae and consumes coral wastes from within and protects the coral from Vibrio and other bad bugs.
Okay. And so what's this second one all about? What is its purpose?
The number 2 is actually also Rhodopseudomonas palustris, but I wanted to point out that they form these things called rosettes.
Oh, right. Okay.
So does it look like a big ball
Yeah.
So they attach to themselves at their polar ends, and they make like these big chunks or whatnot, which is kind of cool because when they're in the plankton, that makes them a bigger size, so it makes them more available to a broader variety of bacterioplanktivores, different types of filter feeders. And then number 3 is a lot of those rosettes connect together, and then they produce biofilms.
Okay.
When you get to that point, that's when you're going to have a lasting population of these guys in your tank. So if you have some marine pure or something like that, you saturate it really well so that they can get in there, and they can maybe beat out other bacteria by number, and also they create that zone of inhibition. It's what they call when when there's a lot of cells They can produce enough antibiotics that it isn't diluted, right? Then it's more concentrated and it kills off other stuff around them. And that's when they make the biofilms, and then they're more lasting. And that's what not everybody sees in their tank. Because again, like we were saying, they don't always survive long-term in the aquarium. It depends on the aquarium conditions. But if you promote their growth, they'll eventually form those biofilms, and they become much more resistant after that because they're really strong in numbers. So the young ones, the new cells are the biofilm, those mature cells are called mother cells. It's the daughter cells that are motile. So they're the ones that bud off, and they're flagellated, and they swim around in the water column. And then they become an in-house, an in-tank source of food for your corals. So they're literally feeding your corals and sponges, bivalves, tube worms, things like that 24/7. The next one I think is Rhodospirillum, and that was the number 2. So I think that's slide number 4.
Yep.
That's the corkscrew-looking guy.
Yep.
Not as good of an image because Rhodospirillum's hard to photograph, get a good resolution because they're corkscrew shape. You know, they go in and out of field so much. But obviously, so Rhodospirillum just means rosy spiral, referring to the color of that bacterium. Those guys are pretty cool because they spend a lot of time in the water column. So they're particularly good as a planktonic food. And they have their own different types of antimicrobial substances that they produce. When you use those with Rhodopseudomonas in tandem, they actually have their own niches. There's a paper, they looked at the populations of those 2 species on a Caribbean reef, and they found that they actually inhabit different parts of the reef. Rhodopseudomonas occurs closer in, and Rhodospirillum occurs more outside the reef crest. So it's kind of cool that they, you can put those both in a tank, and then they'll find the niche that, you get at least one of the 2, right? It increases your chances that at least one of the 2 will survive depending on the conditions in your tank. I think the next one then is I actually have a slide.
It's a wide shot of the same bacteria. Yeah.
Oh, yeah. So that one is the one that just shows Rhodospirillum, but how huge it can get.
Okay.
So that doesn't form the rosettes, but because it gets so freaking huge, really long anyway, it makes it a nicer bite size for bacterioplanktivores when they capture it. Just because they can get to be such long cells, and again, they spend so much time in the water column.
Gotcha.
I think the next one then is actually that's a rosette of Rhodopseudomonas palustris.
Number 6.
Yeah, there's like another big ball, right? A rosette. So that's Rhodopseudomonas, which is the one in ProBio, and then it's got the spiral one next to it. Yeah. Which is Rhodospirillum, and that's the one that is in addition to Rhodopseudomonas in substrate sauce, the cycling product. So just for size comparison, right? The rosette's like a big ball, but the Rhodospirillum is super long and windy. So again, like it gives a lot more opportunities to filter feeders because some of them are pretty specialized in how they capture particles.
Yep. And the next one, number 7, is spheroids.
Spheroides, yeah. So that's
not steroids.
So that's Rhodobacter.
Okay.
Rhodobacter is a massive, massive family. Not all of them are purple non-sulfur bacteria. In fact, some of them are notoriously evil, and some of them are really, really
evil bacteria.
Evil to corals, and then some of them are actually like critically important to corals ecologically. So it's a huge varied family. Again, not all of them are even photosynthetic. It should be split actually, that family, into maybe 2-3 different families. But anyway, so that's Rhodobacter. Originally we thought it was a closely related species. Originally I was calling that Capsulatus, but part of that had to do with, so again, like I mentioned, these guys grow, they have different shapes sometimes depending on how they're grown. And then actually Aquabiomics helped resolve that one for me. Because again, that's one of the species that we isolated, very closely related, spheroides. But the difference is very significant because originally I was just selling that in the kits, the home grow kits, to freshwater people. Because capsulatus is pretty much exclusively freshwater. But that was really exciting to do it. We did a test growing it in after I got the Aquabiomics test back and found out it was actually spheroides. I grew it and I tested it in salt water, and it actually grew really well. And it's quite beautiful. So that's really nice because I know I haven't studied it much, but spheroides also produces compounds that are known to inhibit Vibrio. So there are implications with that species for, again, like RTN and other such diseases.
Gotcha.
Just not something we've looked at really closely yet.
And the next one, the denitrificans close-up?
Yeah, that's Rosiobacter. So that's
okay.
Yeah, that's the only species in these slides that I don't sell. I have cultivated Rosiobacter, but it's been really challenging to grow on a commercial scale. That's the only purple bacterium that I've grown that is actually aerobic. But that one is massive. That's actually in Rhodobacteriaceae or whatever, the Rhodobacter, same family. But that one's huge. It's like in some environments, the Rosiobacter clade makes up like 20% of the biomass of some marine environments. It's just huge. As its name implies, it's a denitrifier. But one of the things it's really good at is just metabolizing DMSP, which I was mentioning earlier. Remember, that's a substance that corals release when they're stressed.
Right.
I think so. Again, like if you were trying to prevent Vibrio from attacking corals, that would be another really good one. But just in terms of like cleaning the water and acting as a real nice probiotic.
Gotcha.
So we're looking at that one, but just haven't had success. So not good at growing all of them.
Yeah, learning process. So the last one is an unknown one.
Yeah. So this month it'll be a year. We made a expedition to Polynesia, and we collected samples, sediment samples and coral mucus samples from 3 different sites, all Porides, healthy Porides colonies, like great big, like, half a Volkswagen car kind of size boulder, and had them analyzed for the microbiome just to see what was in them and what the reef looked like. And I also wanted to bring home some bugs as souvenirs and see if I could cultivate them, see what I could get. And it turns out that there were no Rhodopseudomonas there. There was no Rhodospirillum there. Why? Well, the reason is, so going back to, like, people say, these bacteria won't grow in my tank, so why would I use them? So it's so we spent most of our time on Moorea, and actually spoke. If any of you know Coral Gardeners, they are a coral restoration group there, and I met with their head biologist, and she was explaining to me why there was all this algae growing on the reef. The reef is actually somewhat degraded at this point because of pineapple plantations, and because of the fact that they probably treat less than 10% of their sewage. So the water is pretty eutrophic. There's a lot of nutrients in the water. I wish I should have brought that with me. I can't remember. We did all the water tests, but the nitrate was comparable to what you'd see in an aquarium, rather than a healthy natural coral reef, where we're talking about, on a coral reef, nitrate actually on average, according to one paper I looked at, was like 0.1 to 1 micromole.
Yeah.
Very low. We're talking so that's like 0.06 to 0.006 parts per million nitrate. There's no need to add nitrate to your reef tank unless you're trying to grow algae. One of my gripes, another story, I won't even go there. But so that you shouldn't have nitrate. You don't want nitrate. Susan Telly can't even use nitrate, or at the very best, they prefer ammonia to nitrate, which is why nitrogen fixing bacteria are important. But anyway, so we went to this reef, tried to collect the bacteria I want, or at least see that they were there. I wanted my own strains of Rhodopseudomonas or whatever, and they weren't growing there. Well, that's interesting because they should be there, but because of all the nitrate in the water, other types of bacteria are out competing those bacteria. Right. So further north in Polynesia, there was a study done actually looking at Rhodopseudomonas on a reef that had been polluted. I actually found this after the fact online, but they looked at a Hawaiian reef somewhere that had been polluted, and they found Rhodopseudomonas growing per normal offshore, but in that immediate area, I don't know if it was outside some golf courses on Waikiki or whatever. I don't know. I can't remember where in Hawaii, but where the nitrate was elevated, those Rhodopseudomonas was absent or scarce. And the reason wasn't that they can't live there. They can use nitrate themselves, but they have a distinct advantage over other bacteria in a nitrogen limited environment because they can fix nitrogen. They don't need nitrate or ammonia in the water. They just take nitrogen gas, right? The atmosphere is 80% nitrogen gas. It's there for the taking, but you just have to be able to turn it into ammonia, and very few bacteria can do that. So if you remove those from that environment, then you're also removing the benefit those bacteria have to the corals. And all of a sudden you have all this nitrate in the water. You're also encouraging, there was sargassum growing all over. I didn't even know you'd see that in Tahiti, right? I always pictured it being like crystal clear water, and the water is more or less clear, but there was so much benthic algae. And the corals really were hurting. You could see that.
Interesting.
Yeah, so learned a lot there, but I did bring back some incidentally bacteria that would be more appropriate for the typical aquarium, because the water there was more like the typical aquarium. So I grew those in, and this is relating to that picture you're referring there, but I grew samples of sediment in, I put samples of sediment in growth media, the same growth media that we use to grow PNS substrate sauce. So again, a saltwater growth medium for heterotrophs. And I was able to grow various types of phototrophic bacteria. So it looks like we have, so we had the initial cultures looked at by Aquabiomics, but now that there's a process where you eliminate things, and we're getting down to where we have, it's kind of cool. There's a co-culture. So two bacteria that sometimes you'll have like what are called guilds. And this is important for people that use bottled products. Sometimes, you get a single species in a product and they may not persist because they rely on other species to survive. They don't always compete. Sometimes they work together to survive in groups. So in this case, I try to get like a functional group of bacteria, a guild, if you will, to live in this medium. And now it's going on a year. I've just been making successive cultures, and whatever can't live in it has just been eliminated, and I can't go back to Tahiti to get it. But what we have left now is a purple sulfur bacteria, not non-sulfur, purple sulfur bacteria. So they're the ones. By the way, if you're wondering why purple non-sulfur bacteria, that's a stupid name, right? But it's just because purple sulfur bacteria were discovered first, right? They're the ones that eat hydrogen sulfide, right? So the purple sulfur bacteria are similar, but they don't have that ability. In fact, like most other things, they can't actually handle a lot of sulfide. But purple sulfur bacteria like it because they actually use it as part of their metabolic processes. So anyway, we have those guys in there, and when we have green sulfur bacteria that also metabolize metabolize sulfide. So why is that important? Well, if I can continue to cultivate these bacteria, we will have a natural, wild coral reef bacteria. A lot of people that are just for they're like soil bacteria, or they're just fresh water or whatever. These are from an actual coral reef. And these particular bacteria are applicable for uses in, say, not everyone has a deep sand bed in their aquarium, but maybe they have one in their refugium. or maybe they have so much rock in their tank that they actually have anoxic pockets where you can get hydrogen sulfide. Well, these particular bacteria would actually consume that hydrogen sulfide. So just another thing that you can add to broaden your microbiome and make something more close to nature.
There's a lot of ingredients you could add to the pot there, so to speak. Fascinating stuff, dude. It's really quite a. I never had an idea that you'd actually go on a mission to collect certain bacteria across the world. So that's pretty neat. Kind of blows my mind. So Kenneth, man, I think we're gonna wrap up the live stream. Any final thoughts before we close this out tonight?
Yeah. First of all, I'd like to thank everybody who supported us so far. I wouldn't be able to sit around reading about bacteria all day, which is actually something I like, if it weren't for all you people that support us. So I appreciate that very much so. And to any manufacturers out there, actually, I'd say, reach out. This is what I do. I cultivate bacteria, but there's so many. For example, anybody that makes equipment, there's room for a plankton reactor, a photosynthetic plankton reactor. Julian Sprung wrote about those possibilities like 18 years ago, right? With Rhodopseudomonas making a reactor for something like that. And nobody's tackled it since. So, I'd be happy to work with someone who would want to collaborate on something like that. Foods. These are, by the way, like a good fish food or a fish probiotic. They occur in nature in fish guts, including saltwater fish. They've been shown in captivity to increase disease resistance and growth rates, things like that. So, I mentioned earlier here that they can be frozen. So, anybody out there that manufactures foods, if you wanted to add a probiotic, you can reach out to us for something like that. But yeah, in addition, just to everything, all the anecdotal evidence that our supporters have provided, anything that I could do to work with other manufacturers to kind of improve how these bacteria are used would be awesome.
And hobbyists can get your home grow kits on your website, hydrospace.com. Is that the web address?
It's our web address, but we don't visibly advertise that product since I pulled we don't actually have a store now that we have a distributor.
Okay.
But you can get it through the back door if you just Google PNS Home Grow. So that's PNS space Home Grow, just GRO. And you can order it that way. It'll come right up. And by the way, if you're an LFS, you can get our products from we have an exclusive US distributor, and that is Aquarium Supplied Distribution, ASD. Chad and Orlando are really great guys if you haven't met them at a show or something yet. So I'd encourage reaching out to them, not just for hydrospace products, but for all kinds of stuff. And then stores, there's many places online, and then maybe some of your favorite LFSs already carry it, but Bulk Reef Supply, saltwateraquarium.com, Premium Aquatics, more recently Unique Corals, Champion Lighting. A lot of people have it available now. Yeah, so
cool.
At retail.
All right, Kenneth, like I said, man, fascinating stuff, and always a lot of nuggets to digest tonight, but appreciate all the insight and the expertise from you. And also want to thank both Bulk Reef Supply and Ecotech Marine for supporting the live stream, sponsoring the live stream, and all you folks out there for tuning in. And I also want to give a big thank you to Paul, who is the moderator as well as the president of the Boston Reefer Society. Please join and support your local reefing clubs. They are so important to this hobby. Also want to let you know that all episodes of Rappin With ReefBum are available as podcasts on Spotify, Apple Podcasts, Google Podcasts, Stitcher, and Amazon. My next live stream is actually going to be a live coral sale on YouTube this Thursday at 8 p.m. Eastern Standard Time. I got some kick-ass premium SPS frags that are going to be up for grabs, including a couple of some rare Tyree Purple Monster frags. So I know got some folks pining for those frags. My next Rappin With ReefBum live stream will be next Tuesday, October 10th at 7 p.m. Eastern Standard Time with Adam Sutherland from Frag Garage. So that should be another great show. If you want to check out the full upcoming schedule of guests, visit reefbum.com under the YouTube section. Until next time, be safe and be well. Later.